Thank you, everyone, for joining us today. Before we begin, I wanted to begin with some webinar logistics. For this webinar, all lines will be in listen-only mode. You may submit your questions at any time using the Q&A or chat box and select all panelists. You may need to activate the appropriate box using the floating navigation panel, and this can be found on the center of your screen, and it looks like this shown here. And you have to click on the little bubble icon to expand the chat and Q&A box. Closed captioning is available by clicking the link in the chat box, and note that this webinar is being recorded. So, thank you. Today's webinar is about the participant engagement in cancer genome sequencing network funding opportunities, and we'll be introducing these funding opportunities today. So I'm Leah Mechanic. I'm from the Epidemiology and Genomics Research Program in the Division of Cancer Control and Population Sciences. I'm presenting on behalf of Dr. Galanders and Dr. Cho, the other scientific contacts on these funding opportunities. Dr. Galanders is also a program director with me in the Epidemiology and Genomics Research Program, and Dr. Cho is from the Behavioral Research Program. Our expertise is complementary for these funding opportunities, and we will work together to address your questions. We will host 2 pre-application webinars for these funding opportunity announcements. These funding opportunity announcements are associated with the Cancer Moonshot initiative that is intended to accelerate cancer research. Specifically, these funding opportunities fall under a scientific priority designated by the Blue Ribbon Panel as Recommendation A, to establish a network for direct patient engagement. Today's webinar will focus on introducing both funding opportunities, and the second webinar in September will focus on answering the frequently asked questions for both of these funding opportunities. Following these webinars, we will post a recording for each webinar on the website listed. Please note that while we will be talking about these funding opportunities, we will not discuss any individual investigator's specific aims. Here's the outline for today's webinar. I'll give a background behind these funding opportunities, talk about the participant engagement in Cancer Genome Sequencing Network, or PE-CGS. We'll talk about the first funding opportunity for the research centers. Then we'll talk about the second funding opportunity for the coordinating center. And finally, we'll have time for questions. So first, the background. Genome characterization efforts, such as the Cancer Genome Atlas, have been very successful, but research gaps remain. On this slide is an example of one such research gap. Looking at the race distribution of tumors from TCGA, only 14% come from minority populations. If you look at the ethnicity distribution of tumors in TCGA, only 3% come from Hispanic or Latino populations. To quote a recent commentary, results such as these have resulted in substantial gains in the understanding of cancer prevention and treatment, yet their generalizability to all U.S. populations is limited due to the lack of racial and ethnic diversity. It's imperative that the Cancer Moonshot not repeat this history. One method to address these research gaps is a direct participant engagement approach. And shown on this slide is an early example using this approach for a study of myeloproliferative disorders. The investigators here used an internet-based protocol to collect clinical information and biological specimens for several hundred patients. They determined that a significant number of patients had recurrent mutations which were a potential target for pharmacologic inhibition. This example highlights how the internet can expand our reach beyond traditional clinic-based approaches and address a research gap, and in this case, a study of a rare cancer. For the purposes of this funding opportunities, we define participant engagement as an ongoing, bidirectional, and mutually beneficial interaction between participants and researchers, where participants are included as an integral part of all phases of the research process, including the identification of research priorities and the design, conduct, and uptake of research. By direct participant engagement, we mean that research teams interact directly with participants using the web, social media, or online patient communities as examples, not necessarily through providers or the clinical care setting. We also mean you incorporate input throughout the research process through surveys, interviews, and regular communication from consenting to return of results. So next we'll talk about the Participant Engagement in Cancer Genome Sequencing Network, or PE-CGS network. The NCI intends to support the PE-CGS network, which will include several U2C research centers and one U24 coordinating center. The network will also include a steering committee and a subcommittee to the steering committee as an external advisory panel, and this is described in the RFA. The network will function as a collaborative network, allowing the research centers to address common issues, share best practices and lessons learned, and utilize common methods where appropriate. The activities funded for this network must address the Cancer Moonshot Open Access Pilot Program. Using the provision outlined in the 21st Centuries Cure Act, NCI has established a data sharing policy for projects that are funded as part of the Cancer Moonshot that requires applicants to submit a public access and data sharing plan that, one, describes their proposed process for making resulting publications and, to the extent possible, the underlying primary data immediately and broadly available to the public upon publication, and two, if applicable, provides a justification to NCI if such sharing is not possible. The NCI will give competitive preference and funding priority to applications that comply with the strategy described. The data sharing will become a term and condition of the award. You can find more details at this website. So the goals of these funding opportunities are twofold. First, to promote and support direct engagement of cancer patients and post-treatment cancer survivors as participants in cancer research. And second, to use such approaches for rigorous cancer genome sequencing programs, addressing important knowledge gaps in the genomic characterization of tumors in areas such as, but not limited to, rare cancers or rare cancer subsets, highly lethal cancers, cancers with an early age of onset, cancers with high disparities in incidence and/or mortality, or cancers in understudied populations. So now that I introduced the network, I'll talk about the first funding opportunity for the research centers. I'll provide an overview and describe the structure of the U2C research centers, provide some application details, and talk about the administrative core and functional units for these centers, provide an overview and points to consider. So for the U2C research centers, the application due dates are October 30th and July 30th, October 30, 2019 and July 30, 2020. The letters of intent for these applications are optional and they're due 30 days prior to the due date. And these letters will help NCI in the planning for the review. We intend to commit $12 million of total costs in FY 2020 to fund up to 3 awards, and note that these could be selected in the first round. Application budgets are limited to no more than $2.5 million per year in direct costs and need to reflect the actual needs of the proposed program, and the maximum project period is 5 years. Each proposed research center should be centered on addressing a unique knowledge gap in the genomic characterization of tumors. The knowledge gaps proposed for the characterization are expected to be mainly identified in the specific areas of interest, which were the areas I described earlier as priority areas. Nonetheless, if there's a compelling justification available, knowledge gaps outside of these areas may also be proposed as interest area 6 with a strong justification. Some points to consider for the research center. The multidisciplinary team should include an appropriate combination of specialties, include social and behavioral scientists, oncologists and pathologists, molecular biologists, geneticists and bioinformaticians, and genetic counselors. And each research center will be expected to collaborate across the network with the other research centers. These collaborations will be facilitated by special set-aside collaborative funds, and the collaborative projects are intended to facilitate the sharing of best practices, the optimization of approaches, and addressing common issues. So each research center must have the following components. It must have an administrative core and 3 functional units: the participant engagement unit, the genome characterization unit, and the engagement optimization unit. So you'll write your application addressing these, the core and the 3 units, as well as you'll have an overall section. So in this slide are the page limits for the research strategy sections for these different components. You have 12 pages for the research strategy for the overall and the 3 functional units, and 6 pages for the administrative core research strategy. Each of the sections that I talked about has its own required research plan structure. So take a look at the funding opportunity for details. For example, the overall section requires these listed subsections: research focus and significance, research center organization and team integration, overall approach and structure of the research units, milestones and timelines, network collaboration, and health disparities. I wanted to point out the milestones and timelines because this is part of all the sections, and they also refer to as benchmarks or progress evaluation, and you must include in this section timelines and description of how you'll achieve your goals. These must be for both individual participants and outcomes for research. Note, be sure to also speak within the application to the significance and innovation of your approach, and be sure to review the RFAs for further details regarding the structure and the review criteria. So one thing that is different about this funding opportunity is that there are several required attachments to be provided in the overall section. These include Attachment 1, a table listing of available partnerships and infrastructures along with their main characteristics, and you'll use the file name Partnerships and Infrastructure. Attachment 2 is the U2C Research Center Engagement Process. We'll use the file name Engagement Process. And the third attachment is Example Draft Consent Forms. So now I've talked about the overall application structure. I'll go into details about the administrative core and the individual functional units. The administrative core will provide the administrative support to the U2C Research Center leadership. The main roles of this core will include oversight and support of all research activities, facilitating interaction among the 3 functional units at the center, and it will support coordination among the U2C research centers and implementation of agreed-upon network practices and principles. So the first functional unit is the Participant Engagement Unit. This unit will be responsible for the direct participant engagement for the purposes of performing genomic characterization. This unit will direct participant engagement, recruitment, tissue acquisition, perform data collection, and return of information to participants. The number of participants will be justified by the cancer frequency and the characteristics of the cancer. And there will be a critical role in this unit for oncologists, pathologists, and genetic counselors. So there's some points to consider for the Participant Engagement Unit. The participant engagement strategies proposed are expected to use current state-of-the-art, culturally sensitive approaches. This unit should be designed to ensure continuous improvements in the engagement process by interactions with and adapting research findings from the Engagement Optimization Unit. You see the RFA for further details regarding activities of this unit, including the reporting of research findings to participants, as well as returning individual-level genetic information to those participants who are interested in receiving such data, and maintaining an ability to recontact participants to obtain follow-up clinical and/or epidemiology data. Also note that blood specimens should be proposed as the source for normal DNA. However, in situations where it's not possible to collect blood specimens, an alternative source may be proposed if appropriate. The return of individual results is a critical way to engage and respect participants. Therefore, in these centers, participants need to be able to opt in to receive, depending on their preferences, summary of individual health information used in the study, ongoing study updates, aggregated results, lay summary of scientific findings and research results, individual germline and somatic genetic results, and other information preferred by participants. Performing the sequencing in a CLIA laboratory will allow for broader return of genetic information. For this network, we have a recommended germline return policy where the center will report back to participants and their oncologist pathogenic variants within the ACMG59 cancer genes. There will also be a recommended somatic return policy to report back to participants and their oncologists potentially actionable variants. Additional plan details relevant to the specific cancer type or population study can be suggested by the U2C investigators or applicants. And it is anticipated this area will evolve and this policy will be periodically evaluated and revised. So the second unit is the Genome Characterization Unit. This unit should be capable of completing all the steps required for the tumor and normal characterization, i.e. the DNA and RNA sequencing, bioinformatic analyses, and the interpretations. The sequencing is to be performed in a laboratory certified and fully compliant with CLIA to allow for the return of genetic information. The required characterizations are whole exome sequencing, RNA sequencing, and low-pass whole genome sequencing. No, applicants may propose additional types of genomic characterization if appropriate for the cancer they're studying. And the data will be submitted to the NCI Cancer Genomic Data Commons in a manner consistent with the informed consent. Note the points to consider for the genome characterization unit. First, we're requiring that sequencing will be done in a CLIA laboratory so that genetic results may be returned. Because this is a research study and not a clinical sequencing study, applicants should propose to batch sequencing to maximize efficiency. Next, since this information will be reported back, applicants must consider the need for an FDI investigational device exemption and discuss this with their IRB. And finally, each research center is expected to share information about the algorithms used for interpretation and will be encouraged to share informatics tools. So the last functional unit is the engagement optimization unit. This unit should be able to incorporate rigorous behavioral research on optimal approaches to participant engagement in various aspects ranging from recruitment, communication, to education about genomic characterization goals and discoveries. The interventions to be developed and tested should be designed to identify optimal approaches. For example, directly reaching and communicating with potential participants about the goals and values of genomic characterization, engaging with understudied and/or underrepresented populations, using web-based and social media platforms, and leveraging established partnerships with patient communities. So some points to consider about the engagement optimization unit. The research activities and interventions to be developed by the engagement optimization unit should be fully integrated and serve the overall goals of the research center. The applicants must identify the research questions and participant engagement and provide plans to address these questions in this unit. And the applicants must describe how the results from the engagement optimization unit will inform the protocols developed by the participant engagement and genome characterization units. So on this set of slides, I want to try to put this all together so you see how the functional units interact. So the participant engagement unit will use innovative approaches to capture participant preferences and inform study design. They will consent participants and provide an opt-in consent for the return of data and results. The participant will grant the center permission to collect tissue samples and clinical data, and the center will collect epidemiology data. The participant engagement unit will provide regular updates and be in communication with participants. This participant engagement unit oncologist and genetic counselor will return to the participant and their oncologist the genetic results. The second unit, the genome characterization unit, will be responsible for the CLIA sequencing pipeline, the analysis and interpretation, and the identification of those variants to include in the report, both germline and somatic variants. And this will be provided to the U2C oncologist and genetic counselor, and the results will be provided to provide study updates. The third unit, the engagement optimization unit, which is the unit that will be performing the empirical research and participant engagement, will ask questions relevant to the other units. And shown in these green boxes are examples of the types of questions that this unit may address. For example, this unit may look at the top left box, methods of reaching and recruiting participants, which could inform the recruitment of participants. They may also ask questions about the communication of study goals, which can inform the design of the study consent. They may also ask about effective and participant-centered approach to returning research results, which would inform how these results are returned. And they may also address questions about understanding the preferences for return of results, which could inform the design of the report that should go back to the participants. Note that the participant engagement unit and the genome characterization unit will start with an initial protocol based on what we best know now, but they should be able to iterate their protocols based on lessons learned from the research in the engagement optimization unit. So the second funding opportunity is for the PE-CGS Coordinating Center. I'll go through an overview and structure of this U24 Coordinating Center, describe application details, and provide some points to consider. So the Coordinating Center applications are due October 30, 2019 and July 30, 2020. We request that 30 days prior to the due date that you submit a letter of intent to help us with planning. Note that if an awardee is selected in the first round, this funding opportunity will be expired. We intend to commit $500,000 of total costs in FY20 to fund one award. And application budgets are limited to no more than $350,000 direct costs and need to reflect the actual needs of the proposed project, and the maximum project period is 5 years. The PE-CGS Coordinating Center will operate as the supporting infrastructure for the network. It will collaborate with the U2C Research Center investigators and interact with NCI staff members and with other stakeholders as needed. The major responsibilities of the Coordinating Center will include administrative and scientific coordination of network activities, or network coordination, the development and coordination of outreach and promotion materials and communication channels, the outreach and promotion, and the development and implementation of standardized approaches to data collection and processing, as well as other best practices for the network, or network best practices. So see the funding opportunity for more details about the specific functions. Some points to consider for the Coordinating Center is that the composition of the research team must ensure the requisite expertise and participant engagement in cancer genome characterization. The team should be capable of effective communications and productive collaboration with various stakeholder groups. In addition, the team is expected to include senior members with considerable leadership skills and experience in coordination of multi-center networks with large research teams and/or multi-stakeholder groups in diverse settings. And finally, the U24 Coordinating Center must have experience in development of accessible, user-friendly communication materials and the ability to leverage various communication channels to share information to target end users. For the application for the U24 Coordinating Center, in lieu of the standard subsections, the research strategy must consist of the following modified subsections. Subsection A is the vision and organization, B is network coordination, C is network outreach and promotion, D is network best practices, and E is health disparities. Be sure to also speak to the significance and innovation of your approach in your application, and note in the funding opportunity specific review criteria and cooperative agreement terms and conditions of award. In summary, this network will address research gaps in the molecular profiles of cancer. It'll determine the effectiveness of a direct participant engagement approach. It'll provide insights into development and sustainability of a larger network for direct participant engagement by learning and responding to participant preferences and identifying optimal methods of engagement and communication. And finally, the data will be shared as a resource for the research community. And with that, I want to thank you, and we're happy to take any questions now or more questions later by email.
Okay, so you're free to continue to submit questions using the chat box that Leah described, and I'll just go ahead and start with a few that we've received already. The first question was why epidemiologists were not listed in the key personnel for the multidisciplinary teams for the research center's FOA. So I guess as a genetic epidemiologist, I can say that that was just an oversight on our part in terms of not listing it on that list, but hopefully it was clear in Leah's presentation that we're expecting the research centers to collect epidemiologic and clinical data, and that we see that as really a critical part of this RFA and something that will be different from previous genome characterization efforts. Okay, a second question was what what counts as cancers in understudied populations?
Yeah, so that's a good question. As we noted in the funding opportunity, understudied populations and a specific group who may or may not be underserved or socially disadvantaged, but whose data on cancer health risks and outcomes are currently limited. And there's a lack of sufficient data on this diverse group to inform evidence-based cancer control, prevention, and intervention. So there's a dearth of research and information for these groups that underscores the urgency to improve our available knowledge regarding cancer risks and outcomes within these understudied populations. So as long as the cancer fits or cancer type fits this definition, this would be considered understudied for this funding opportunity.
Okay, and I should have introduced myself previously. I'm Liz Glanders, and I wanted to actually just draw your attention to the email address that is on the screen now, because we're getting lots of questions, and I don't anticipate we'll get to answering all of them. And we also are going to try to keep today's discussion really at a very general level. And so if you have specific questions, feel free to email use this email, and Leah, Sylvia, and I will be monitoring that email. Another question was, will the PowerPoint slides be made available? And the answer to that is yes.
Yes, and we'll be posting a recording of this presentation as soon as possible.
Okay. Another question is, where can we find the RFA to review? And so the answer to that question, if you just Google
NCI PE-CGS, you can find it, or RFA-CA-19-045, and you'll find the links to both of them.
Yeah, so in the 19-045 is the RFA for the research centers, and it has a link to the accompanying coordinating center.
Right.
Yeah. Another question is, are these funding opportunities for established cancer centers or NCI designated cancer centers?
I think that the funding opportunities are open to anyone who can apply, who could put together the appropriate team.
Sorry, thank you all for the great questions. It's just taking a little while to read through them. For the centers, a total of 3 grants or 3 research centers will be funded in the 2 cycles combined, question mark?
Yeah, we're anticipating that a total of the 2 cycles combined.
Is the U24 page limit the standard 12 pages?
Yeah, so it is the standard page limits for the U24.
Okay. What is meant by outcomes for participants in addition to research outcomes?
Yeah, so participant outcomes are those outcomes, including the participants' perspectives on whether they felt respected, empowered, and motivated to participate in the research program. And research outcomes include such things such as the status of sequencing analysis and the analysis of data. And so we see the participant outcomes as a metric for evaluating a meaningful engagement with the study participants.
Okay. If I want to do another type of genomic characterization in addition to the required characterizations. Will those assays need to be CLIA certified?
So it's important to note that the funding opportunity requires specific genomic characterizations with, adequate power and extensive engagement activities, and these should really be the priority of your research application. But if you propose assays beyond the genomic characterizations that we require in the RFA, these do not necessarily need to be CLIA certified. However, these assays, as well as the required genomic characterizations and the extensive participant engagement, must be within in the allowable budget, which is no more than the $2.5 million direct cost per year. And the selection of these assays must be well justified for the cancer type.
Okay. Would hybrid approaches like sequencing in a research lab and validating of actionable targets in a CLIA lab would that be appropriate?
Well,
I guess if that would probably be one that if you had that specific question, it would be helpful for us to talk through the specifics of your application, and you could just follow up with an email to the NCI PE-CGS email. All right, I have then, yeah. Is it possible, one question, one participant asked whether it's possible that genotyping might be done through NCI to reduce costs to applicants. And I think that in general, that would have to be a collaboration discussion that applicants would need to have with, I'm assuming they're talking about intramural NCI investigators. But in general, there isn't a mechanism for us to provide genotyping through NCI unless the applicant had a established collaboration with or a partnership with somebody in intramural NCI.
But this is another case where if you have a specific example and something you're specifically thinking about for your aims, it would be good if you kind of reached out to us and we could talk to you in more detail.
Thank you for continuing to submit these questions. It's really great. Another question was, would community-based cancer care qualify for one of the priority areas? And I think that's probably one question that seems like it would be best for you to follow up specifically with us, because you could imagine it being an understudied population, but I think it would be important to discuss the specifics of your area of interest. Okay. So in participant engagement, are you specifically talking about return of results?
So the return of results is definitely one way to engage and respect participants. and I think we talked about it as an important way. But this is not the only way to engage and should not be the only way to engage with participants. And as we talked about in the presentation, you know, the research center should plan to communicate to participants to opt in to receive several different types of information, as well as to opt in to receive the individual genetic, germline, and somatic data.
Okay, great. So there's a couple of questions that relate to the priority area of rare cancers, and in particular, talking about the sample sizes that are needed. And this is another example where we're going to keep our discussion today general, but if you have specific questions, please feel free to contact us.
So, you know, our thinking had been, you know, this, and one of the reasons we don't give an absolute number of samples is that it's as long as the results are interpretable, as long as we can, you know, make conclusions from the results, I think that is okay, as long as you have, you know, a strong scientific justification for how you set up the study.
And related to that question, I think can we combine different rare cancers to achieve recruitment of the recruitment goal of hundreds of patients?
And I think this comes back to exactly what the scientific justification and rationale for the study design.
Okay. Does the RNA sequencing have to be CLIA certified?
You know.
Yeah, actually, V and I are just sort of looking at each other, and I don't personally know the answer to that. I think the answer is no, just the DNA sequencing. But we will, if this was your question, please feel free to send it to us again, and we'll make sure just to confirm that. So, will this study help patients currently undergoing treatment for cancers or for cancer?
So, you know, while it's possible that the results of this study could help patients undergoing treatment for cancer, the primary purpose of this study is to engage participants, both patients and cancer survivors, in research and contribute knowledge to better understand and treat those cancers in the future. Therefore, it's much more likely to benefit future patients.
Okay, great. Could you say a little bit more, Leah, about the collaboration set-aside funds? How much, what amount should be set aside in the applications, and do applicants need to specify what projects they're going to do ahead of time, and what kinds of projects are you anticipating that they might work on?
Okay. Yeah, so starting, as far as the funds for the collaborative set-aside, this would start in budget period 2 of your application, and that you would put aside a minimum of $200,000 per year in direct costs to be allocated as a restricted fund for the support of post-award collaborative projects between the research centers. And in your application, you wouldn't necessarily have to propose exactly what you would be doing with this collaborative set-aside, but you would have to be proposing or anticipating what some of the common issues might be, or what are some of the common questions and collaborations you might need to do. and describe how you have the flexibility to adapt your project to these collaborative activities. Thinking about thinking about what type of collaborative activities could enhance from the network, could enhance your research center, and how you may work with NCI and the coordinating center in these types of activities. So you wouldn't necessarily have to have it all worked out at the time of your application, but kind of think about being ready and thinking about the types of questions that could be asked.
Okay, great. We did receive one pretty specific question about the genome characterizations that are outlined in the FOA, and so I would just encourage those of you who have questions about that specifically to just follow up with an email to us directly. One question that came in was around the language in the U24 Coordinating Center FOAs that suggests that the Coordinating Center needs to have expertise in participant engagement and cancer genome characterization. So if you could just provide a little more insight into what kind of experience you're expecting.
Yeah, so as far as these expertise, they do not necessarily need to be actively performing the participant engagement or cancer genome characterization, but they should have sufficient knowledge to support the activities of the research centers and this expertise and understanding about both these approaches and these scientific methods. And
perhaps what some of the key issues might be facing each of the research centers in the network, because this will likely help guide the types of collaborative studies that they would be involved in helping to support.
Yeah.
Okay, I think just going back through all the questions we received, I think we've covered all of them, but let me just double check. So there was one question about, this was a question that came in actually during the registration process, and the question was, does the application provide support for resource development as related to the scope of work?
Yeah, so I'm not totally sure what is meant by resource development in that question. We do have in the budget sections under Section 4 of the FOA for the U2C Research Center, we describe the allowable costs. And so I think in trying to understand that, you can look at those sections and to get more information. But for example, for the participant engagement unit, we envision that the cost should include the funds for the outreach recruitment, the engagement of participants, the acquisition of specimens and collection, the tracking, the sample validation by the pathologist, the data collection, and developing the patient-facing materials for results reporting, the participant follow-up, and developing reporting participant-centered outcomes. And the budget request for this unit may include the funding for equipment, software, and informatics resources. So that gives you a kind of a flavor for the types of things that we said were allowable in the FOA, but I would point you to the budget sections of that to get more detail.
Okay, great. So we just have a couple more questions. So one is related to the early onset cancers, that area of priority area. And the question is this only focused on traditionally early onset cancers, or could the application focus on a cancer that is traditionally later onset, yet has contemporary trend with increased early onset cancer, such as rectal cancer?
So to answer this question, yes, I think that is within the scope of the FOA, and also just the critical piece is to keep in mind sort of the rationale behind your cancer selection of your cancer subset. And so even, for the early onset cancers, explaining what we're going to learn and why it's important to study this specific area, and this has a good, like what you're describing here, it was traditionally late, but now is more early, kind of points to that sort of justification as well.
Yeah, great. So there was a comment about sort of following up, I think, on an earlier question or that Leah had answered about the clinical utility of helping patients currently undergoing treatment. And I think it's correct that we are really seeing the primary goals or focus of this study is really, it's a research study to fill knowledge gaps and will likely benefit future patients and not necessarily current patients. So there was a question about each of the subcomponents. So for each functional group, is there a specific aims page that's part of the 12-page limit? And I don't know the answer to that. Do you, Leah?
No, it'd be separate from that 12 page. It'd be the specific aims page would be separate.
Okay. Okay, so each functional unit does have an aims page, or a set of specific aims, but it's separate from the research strategy section.
Right.
Okay. Then the last question is asking about the allowed budget on a per patient basis, and
so we haven't calculated what the allowable budget is on a per patient basis. We just have the maximum of the $2.5 million of the direct cost per year.
And that will include all of the participant engagement activities, all of the genome characterization activities, and all of the behavioral science sort of iterative design activities in the 3 functional units.
Right.
Okay, one question, maybe one or two more questions. So this one is asking, do teams have to have a demonstrated history of collaboration prior to submission?
So, we recognize that these are going to be teams of diverse expertise. And, it's not necessarily required that they have demonstrated this history of collaboration, but the complementary expertise and experience will show the reviewers about the likelihood that your team would be successful. You probably should show how the collaboration is feasible, the feasibility of the collaboration. But we recognize that new and innovative partnerships are possible and probably important for this FOA.
Yeah, that's the point I was just going to add, Leah, that one thing that's unique about these research centers is really that partnership across disciplines in the 3 functional units. And so it's very likely that there'll be new groups forming or we hope there'll be lots of new collaborations made through this FOA. Okay, let me just take one last look at questions. I think we've answered all of them. I just want to remind everybody that if you had questions as you work through your applications, please feel free to use this email address. Also, we'll have a second webinar, which is September 25th. Yep. And that we're going to be focusing that entire webinar on sort of frequently asked questions, and we'll be tracking those as they come in.
Yeah, so we recognize the funding opportunity just came out, and so that you probably need some time to kind of read through it and come up with additional questions. So we really just wanted to sort of introduce everyone to the funding opportunity, and you can digest it over the next several weeks, and then we'll have time to answer even more questions.
Perfect, okay.
So I guess with that, since we've went through all the questions, I want to thank everyone for participating today, and just to reiterate, you could please email us with any of your questions, and I hope you have a great rest of the week. Thank you.